Okay, let's go to the next step. How likely is it that a whole lot of new randomly generated extra nerve cells would do anything? To do something, they have to become incorporated into the existing circuitry and have a potential to be involved in controlling behavior. Well, this question, I think, turns back really to normal mechanisms of development of the brain. If everything about the development of the brain, the mammalian brain, was strictly regulated, if every neuron that was born was labeled and specific, every connection to it was predetermined, then just throwing a whole new group of nerve cells into the mix wouldn't achieve anything, because they wouldn't carry with them programs for new connections that could be useful. So there must be flexibility in the normal development of any brain if evolution of brains was to be possible at all. So here we are looking at rodent brain, a mouse or a rat brain. Here's the cerebral cortex, which is in humans much the biggest part of the brain, and the part that has expanded most, and the part that's most involved in high-level cognitive function. This is the cerebral cortex. And there are pathways into the cerebral cortex from all of the sense organs. From the eyes, a pathway into a particular subcortical structure, relay structure, and then on up into the back of the cerebral hemispheres. From the skin of the whole of the body, including the whiskers, as shown here, to a different part of the same structure and a different part of the cortex. From the ears to yet another part of the cortex. So there's a system of relaying through this set of nuclei called the thalamus. And this set of nuclei has co-evolved with the cortex. As the cortex gets bigger, this cluster of nuclei here has also got bigger because it's carrying information into the brain. Well, it turns out that what we're asking is, if new cells were put into the cerebral cortex, would they automatically get some input? Could they possibly become functionally organized? And the answer almost certainly is yes, because brain sizes vary in unpredictable ways by small amounts from individual to individual within a species, but they all have to deal with the problem of connectivity. You don't find animals that by chance have a slightly larger brain with large numbers of nerve cells that are not innervated and connected. So the general developmental plan ensures that connections are provided for all the cells that are available. And one of the reasons is the strategy that's used to form the connections. I'm going to describe work done by Zoltán Molnár in my lab some years ago now, in which we looked at the way in which connections are formed between the very early developing cerebral cortex and the very early developing thalamus here. The fibers carrying information into the cerebral cortex come from this structure. And into this structure come signals from all of the sense organs. So that's a crucial inward pathway to the cortex. And it turns out that the connections that are formed are generated before this structure receives any input from the sense organs. So it's an entirely autonomous process of formation of connections between this structure and the cortex before any signals arrive from the sense organs. And the way that it's done is this. If you look at early stages, and this is a method that uses a fluorescent dye which spreads through nerve fibers. You can see it running down here, even in fixed tissue. The tissue doesn't have to be alive. So you can use this to label developing pathways, even in very young embryos, by fixing the embryo, applying a tiny crystal of one of these dyes to one structure, waiting for a time for incubation, and then looking at the way in which the dye has infiltrated axons, so you can see the pathway of fibers. So here, at very, very early stages in the rat, already there are fibers descending from what will become the cerebral cortex towards the junction of the cerebral hemispheres and the diencephalon, where the thalamus is. Now, at this stage, by the way, the cells that are making these axons are the very first generated nerve cells, produced locally from stem cells here in the base of the cerebral wall. They migrate upwards and lodge at the top here. before the beginning of the formation of the true cerebral cortex. These are very unusual nerve cells, most of which die later on in development. They are there as a kind of temporary developmental device to help to form connections. And here they are, sending nerve fibers downwards. If we now do this experiment in which we put two different dyes, one into the cortex and one into the corresponding region of the thalamus having these orange-red labeled fibers, the cortex having green fibers, you can see that they meet each other here, and the thalamic fibers pass over and climb over the sort of lifeline of fibers that's been thrown down by the cortex. So the cortex presents to the incoming, ingrowing thalamic fibers a large array of fibers. The thalamic axons spread themselves over all the fibers that are available and simply fan out and fill the whole of the cerebral cortex, however small or large that sheet of tissue is. So it's a very flexible, adaptive, developmental mechanism. So you can imagine if there are more cells than usual, as a result of an evolutionary change, a mutational change, they will simply fall into place in this flexible developmental program. So how then do the incoming fibers distribute information to the cortex? This is a view of the human cortex. And I'm going to show you the major committed specialized areas for the control of movement here. This is the so-called motor cortex, motor strip, which sends fibers down into the spinal cord to control movement. An area directly behind it, the postcentral gyrus, which receives information from the body surface and from the deep tissues. A visual area at the back here, and an auditory region in the temporal lobe. That's the main distribution of the sensory areas, each of these corresponding to the target of one of those nuclei in the thalamus. Interestingly, this basic pattern of organization was established very, very early in vertebrate evolution, pre-mammalian evolution. If we look at the commonly accepted tree of vertebrate development, going through the common ancestor for vertebrates through to placental mammals, from marsupials and monotremes and on upwards. If we take existing species which are probably representative of the form of the brain during the evolutionary process, let's say a hedgehog, which is an example of an insectivore, just one of the earliest of the placental mammals. If you look at the arrangement of the sensory areas in the hedgehog brain, here they are shown in color on the side of the brain, on the left-hand side of the brain, the red and orange areas being somatic sensory, tactile areas, touch areas, the green one being an auditory area, and the blue ones being visual areas. The basic arrangement of them is not that dissimilar to the human brain, with touch areas here, then auditory, and then visual. And it's the same in all of these mammals. The basic pattern of the sensory area is the same. The huge difference between a hedgehog brain and a human brain is not just that it's enormously larger. This is out of scale. I mean, this is tiny compared with this. But that the proportion of the cortex occupied by sensory regions is much smaller in the human brain. This was already present, this basic pattern, very early. And what evolution has done is to add more and more of this stuff, the stuff in between. It isn't as though you can get from a primitive mammal to a human just by blowing up a balloon, inflating a balloon. It wasn't like that. More and more non-primary areas were being added during evolution.